So good morning, everyone, and welcome to our workshop entitled Models to Better Understand CF Pathogenesis in Biology. This workshop is led by myself and Dr. Finn Hawkins from the Center for Regenerative Medicine and Pulmonary Center at Boston University School of Medicine. This morning, we've assembled 5 excellent speakers, and our goal is to share with you some of the recent breakthroughs in model systems that enhance our understanding of cystic fibrosis and the pathogenesis of it. So our talks are laid out so that we examine some of the advancements made in the induced pluripotent stem cell field, as well as look at some new ex vivo models that enable us to study human large and small airways. Finally, we transition to more models based upon animal studies, in which we look at some interesting models that have been developed both in mice, ferrets, and in sheep. And so welcome to our session. We look forward to a spirited discussion.
Induced pluripotent stem cell, or iPSC technology, has been around for 15 years. These iPSCs provide a powerful starting material to model human diseases in relevant cell types for drug screening and for cell therapy. You may be wondering how iPSC's technology is changing research and drug development for CF. My name is Junjie Lu, and I work in the stem cell team at the CFFT lab. Today, thanks to the organizers for the opportunity, I will present some exciting progress that the field has recently made in deriving functional human airway tissue from iPSCs. There's nothing to disclose. Therapeutics development for CF has relied heavily on materials derived from patient airway, specifically human bronchial epithelial or HBE cells. HBE cultures further differentiated at air-liquid interface as the gold standard for airway epithelium and for CFTR channel function study. HBEs are mainly composed of basal cells, which are the progenitors of the airway epithelium. These beta cells are very well characterized with these important markers, which I will talk about later. However, one critical issue with these cells is that it has been challenging to maintain their progenitor capacity in vitro over time, and therefore limiting their availability and utility. We are also limited in the available genotypes because of how these cells are collected. And this is especially true for rare and nonsense CF variants. And for people with these variants, there is currently no approved therapies. In order to further advance therapeutic development, we need to generate materials that are very similar to these HBEs, and iPSCs are a very promising resource for this purpose. So we could technically take iPSCs from a patient and directly differentiate them to airway, epithelium, and perform drug screening. Could also potentially correct these disease-causing variants in patient iPSCs and then differentiate them for transplantation and cell-based therapies. And this hopefully will become a real one-time cure for CF disease. Before we go any further, it is also important to highlight that there has been a large amount of work in this area before us. Over the past decade, many publications have come out and have helped us understand the developmental processes from early endoderm patterning all the way to increasingly mature functional epithelial cells. In particular, this recent breakthrough from Hawkins and Suzuki et al. Has enabled us to finally utilize iPSC-derived material in CF research. And basically, what we do is based on such collaborative efforts of many labs across many years. What I want to show you here is a simple schematic of the differentiation protocol. As you can see, the differentiation basically follows important developmental milestones and directs iPSCs stepwise through these stages. And this can be done with iPSCs carrying fluorescent reporters of basal cell markers, or with report-free iPS lines. I'll first briefly show you what the differentiation looks like with report iPS lines, such as the one we developed that express GFP from endogenous NKX2-1 locus and mTERI from endogenous TP63 locus. We start from iPSCs, and the first middle step is to reach specification of NKX2-1 positive lung endoderm. Here is shown in green. And this is done in traditional two-dimensional culture format. And these NKX2-1 positive cells are moved into three-dimensional culture with Matrigel to activate TP63, which are further cultivated to bring up NGFR or neural growth factor receptor. At this stage, we have basal-like cells or RBCs that can be continuously passaged and maintained with the ability to further differentiate at air-liquid interface. This entire process takes about 2 months. However, these cells at the RBC stage can be frozen down and brought back again to be expanded. So you don't have to start from iPSCs every time. And to differentiate report-free iPS lines, we would rely on cell surface markers such as CPM in this case, instead of NKX2-1 reporter for the non-progenitor step. We have successfully adapted the protocol and differentiated multiple iPS lines in the lab. I will show you now how we characterize these RBCs and the epithelial tissues they further differentiate into a liquid interface. I will first talk about the surface specification. Do we have the correct cell types that you expect to see in a functional human airway epithelium? Next, we examine if these cells can model CFTR function by electrophysiology, and are they sensitive enough to detect pharmacological responses? Lastly, we explore how these RBCs interact with HBEs by developing a simple co-culture assay to let the cells interface with each other and to test the potential for cell-based therapies. So, surface specification. We first look at the RBCs before further differentiation at air-liquid interface and compare them to HBE basal cells. On the left, these RBCs have the canonical basal markers, including TP63, keratin 5, and NKX2-1. All these match very closely with what you see in primary HBEs on the right. We then put these cells through a liquid interface and ask them to differentiate. We were able to confirm that they indeed produce various mature airway cell types as indicated by this panel of markers, including basal cells, ciliated cells, and different types of secretory cells. We even detected some of the rare cell types, such as this one expressing FOXR1 and high levels of CFTR, indicating the presence of ionocytes. And we then moved beyond individual markers and performed unbiased single-cell RNA sequencing on multiple iPSC-derived ALR cultures and compared it to single-cell data obtained from primary HBEs. We performed the integrated analysis using ThreadV3 and based on a cell type classifier used in Plashat 2018 paper. And we confirmed a very good overlap between HBE and RBC samples, with essentially identical distribution of the 3 major cell types, basal, secretory, and ciliated. And they have similar levels of expression for all these key markers. Again, we also detect those rare CFTR FOXR1 positive ionocytes. Now we have both the correct markers and the correct transcriptome. The next question is, can we study CFTR ion transport function? And the answer to this is yes. And this, in fact, has been one of the most difficult areas for iPSC-derived airway material to achieve. But now, with this protocol, we have seen reproducible CFTR signal by electrophysiology. So when differentiated under the same condition, CFTR wild-type IBC cultures in black and the HB cultures in red produced overall similar CFTR-mediated ion transport activity. These cultures derived from IBCs hold tight junctions and show good transepithelial electrical resistance. We do notice a much reduced INAC signal here in HBE in the IBC cultures, which makes them not ideal for sodium transport studies. And we are actively exploring why and how to improve this. But nevertheless, the CFTR profile is very, very specific, which for our purpose is great and indicates that these cells have the potential to replace the limited primary HBE materials for drug screening. To directly test this possibility, we derived RBCs from a homozygous W12 82X iPSC line and compared it to primary HBEs carrying the same homozygous variant, but from a different patient. We then treated these genotype-matched RBC and HBE cultures with a cocktail of compounds, individually or in different combinations. This include SMIC1 inhibitor, which inhibits nonsense-mediated decay, G418 as a restore agent, and vertex CFTR modulators. We show that the effects of these compounds are overall additive. And amazingly, HBE and IBC cultures showed overall identical responses to these treatments. Now, this also still at early stages demonstrate that IBCs could indeed model CF and have the potential to transform drug discovery for rare and nonsense CFTR variants. The last question that we asked, and this is still something actively ongoing in the lab, is if these IBCs can interface with HBEs and potentially be used for transplantation to rescue CF. The idea is that we know that HBEs alone can differentiate into functional epithelium at air-liquid interface, and I just showed you that IBCs alone can do the same. Now, if we mix them together and ask them to differentiate at the same time in the same well, what will happen? And for this, we again took advantage of the GFP material reporters in some of our IBC lines because they allow us to track the cells long-term via live cell imaging and distinguish IBCs with HBEs. We were happily surprised to see that indeed they can interface each other and coexist in peace. Here we first mixed the reporter RBCs with reporter-free HBEs at different ratios and followed the cultures at multiple time points. At seeding, we saw stochastic distribution of IBCs corresponding to the respective mixing ratios. A few days later, at the time when they were switched to air-liquid interface, we observed clear redistribution of IBCs to small patches and obvious loss of IBC reporter surface areas. 2 weeks later, there were large clusters of IBC-derived cells in the 3-to-1 wells, but the reporters were barely detectable in the 1-to-3 mixing condition, suggesting a difference in the fitness between IBCs and HBEs. We then analyzed the cell fate specification, and for simplicity, I just show you some images from the controls and the 3-to-1 mixing condition. Also, IBC-derived cells are again identified by the reporters, both stained in green here. You can see both ciliated cells and circulated cells are present in the culture. They come from both HBE and IBC lineages, proving that both IBCs and HBEs maintain their differentiation capacity in the co-culture assay. We then asked a more relevant question. Can IBCs rescue CF in this simple assay? By that, I mean if we mix CF HBEs with wild-type IBCs, can we detect the CFTR function? The results are, in fact, very encouraging. We indeed see clear functional rescue of CFTR in the co-culture condition, and the level of rescue correlates very well with the number of IBCs used. And this is very exciting. With that, I hope I have convinced you that these iPSC-derived materials can overall accurately model human airway epithelium with the proper cell type distribution of all similar transcriptomes, the ability to assess CFTR function for drug screening, and the potential to be used for cell-based therapies. Finally, I would like to acknowledge the entire CFFT lab, the CFFT stem cell team, and our close collaborators. Happy to take any questions.
All right, so thank you so much for that very excellent talk. Very fascinating work that we just learned about. And so just a few questions from the audience. One question is, have you noted any differences in the differentiation process or properties of your cells dependent on sex of the donor?
That's a very interesting question. I don't think we've looked at that in detail. Definitely there's line-to-line variations, depends on what the history of those IPS lines. But yeah, that's something that we could further look into.
Okay, excellent. So another question is, I know you said that the mechanism is still being explored, but do you have any speculations about the lack of amiloride response in the CF cells that were differentiated?
Right. Yeah, I mean, this is kind of complicated. We're actually looking, for example, we're looking at the transcription of these different subunits, and right now we don't have a clear clue exactly what's going on. But we have a few other ideas to test to see if we can increase the expression or test the different ratios of different subunits, and use some molecules to see if we can activate if the proteins are there.
Excellent, excellent. So another question from the audience is, are you able to compare CFTR short circuit current in the presence and absence of ionocytes?
The short answer is I don't know. I haven't done that essay yet.
Okay, great. Looks like some stuff to be done. So do you have, so this is a talk question from Ann Harris, and she wants to know, do you have bulk transcriptomic data for your IBC cells?
Sorry, can I repeat that question?
Do you have, so you showed single cell analysis, right? Single cells, but have you done bulk RNA sequencing?
Yes. Yeah, we've done both single cell and bulk level analysis of the IBC stage. So and also we compare it to primary HBEs. They are very close. There's obviously some differences, but they're very similar in terms of the overall transcriptome.
Great. So related to that, so you showed a lot of similarities between the HBEs and the IBCs, but what would you say are some of the main differences?
Like it's a ENaC signal is definitely one of them in terms of the electrophysiological function, and there's just also from the interfacing experiment, that the cell mix experiment that we did, it's clear that there's difference in terms of the fitness. So we're not sure exactly what's the reason of the fitness. There could be a proliferation difference because these cells are cultured in different format, HBEs and the RBCs. And also the different differentiation media could have different effects on these cells. So all these factors could contribute to their functional differences.
Great. So sort of as a related and follow-up question, Scott Randall's asking, can you comment on donor-to-donor variability for your protocols?
Yeah, again, great question. So definitely there's line-to-line variations. We've so far derived more than 10 IPS lines, IPSC lines. There's at the early stage of the protocol, there's different percentages of, for example, AKX2-1 positive cells, or different percentages of the AKX2-1 P63 double positive cells. But once you reach the NGFR positive RBC stage, these cells are quite stable. They're functional-wise, they're quite similar. So it's really the early steps that there's more differences.
Okay, great. So another somewhat related question is from Etzer Vladar, and they're asking, in the early time points of the co-seeding experiment, do you see the two types of BCs failing to form junctions with each other?
I'll say no, but we did not look at the early time points, but we look at the final stage after air-liquid interface, and they do form tight junction, and they interact with each other.
Great. So another question that I was going to ask is, have you actually challenged these cells with any inflammatory mediators or bacteria and demonstrated that they also show the predicted inflammatory responses?
Okay, this is something that will be interesting to follow up. We haven't done that yet.
Okay, great.
Junji, very nice talk. I wanted to ask, we're going to hear from the next speakers about a variety of other models. So where do you see the iPSC model potentially fitting into, many of the existing models that we use in CF already?
Great. So I think there will be a few different aspects. First of all, this, it's very easy to gene engineer these iPSC cells. So that's one way to use these iPSC-derived cells for potentially cell-based therapy. And also because this is, in theory, should be more similar to the primary HBE materials. So I think that would be more close to, in terms of the function, more close to the sort of the gold standard model. Also, I mean, we right now, because these RBCs derived from iPSCs are sort of can be stably maintained, so they provide a sort of unlimited material for example, drug screening and other functional assays. So this would be, I think would be a great advantage.
Do you have any plans for what's the next big experiment that you think it's critical for continuing to confirm the utility of this model?
So one of the things that we think a lot is this, when we compare RBCs with the primary HBs, we also comparing samples from different donors. So one thing we're doing is that we're trying to compare RBCs derived from the same patients where we have the HBs, so that we have a patient-matched comparison. I think if we can get a good functional comparison between these cells, so that would be good proof that these RBCs are a great model to us to do for CF research.
Okay, great. Any other questions from the audience? Okay, so if there are no further questions, I think we can thank our speaker for an excellent talk and a great discussion, and move forward to the next one.
Hello, everyone. My name is Rihanna Lee. I'm a graduate student at UNC Chapel Hill in Scott Randell's lab, and today I will be presenting a new project where we developed a novel method of ex vivo airway tissue culture to model cystic fibrosis. There are no relationships to disclose related to this presentation. Air-liquid interface, or ALI cultures, have long served as the workhorse to model airway physiology and pathology. ALI cultures form a pseudostratified epithelium containing the major cell types of in vivo airways. However, in vivo architecture, interactions with the underlying stroma and structures like the submucosal glands are not preserved in ALI models. Thus, we sought to develop an ex vivo airway tissue model to overcome in vitro limitations. Airway explant tissues have been studied before, immediately after dissection, or after 3 to 4 days in culture. However, a long-term method of ex vivo tissue maintenance has not been established. Today, I'm presenting a novel method of airway tissue maintenance that preserves ion transport function, mucociliary transport, and SARS-CoV-2 viral tropism for at least 3 weeks. Large airway explants, or LAE, were obtained from first to third generation bronchi, whereas small airway explants, or SAE, were obtained from bronchioles with diameter less than 2 millimeters. The airway lumen was exposed, and the surface epithelia stripped away from the underlying stroma under a dissecting microscope. Airway luminal epithelia was then placed on a gel foam sponge and cultured in DMEM supplemented with 10% heat inactivated FBS. Here's an example of 3 small airway explants on a gel film sponge magnified through a dissecting microscope. On the far right is a picture of large airway explants on gel film sponges in a 6-well plate. To assess how representative this model is of in vivo, we first looked at the morphology of human large and small airway explant tissues after 14 days in culture. We found that both LAE and SAE tissues were pseudostratified and well ciliated. We also saw submucosal glands filled with AB-PAS positive material in the LAE tissues as expected. From this, we concluded that our ex vivo culture system models in vivo morphology for at least 14 days. We next assessed the electrophysiology of human large and small airway explants by measuring short circuit current in oozing chambers. Notably, large and small airway tissues had no measurable short circuit current immediately after dissection. Likely, this is due to injury during the dissection process or long cold ischemic times. However, by 24 to 48 hours, shown in the white bar, a resistive barrier had reformed and the electrophysiology could be measured. We found that human large airway explants maintained electrophysiologic properties between 24 and 48 hours and the 14-day time point. Importantly, we also found that CFTR function in large airway explants was similar to that of ALI cultures generated from the same donor. By contrast, small airway explants had higher CFTR function at 14 days in culture. Possibly this could indicate that SAE tissues were not fully recovered from dissection at the 24 to 48 hour mark. These findings align remarkably well with prior studies of freshly excised bronchial and bronchiolar epithelia. From this, we concluded that large and small airway explants maintain an in vivo-like electrophysiology for at least 14 days in culture. We then wanted to see if human large airway explants from CF and non-CF donors would exhibit the expected differences in electrophysiology. To assess this, we compared CF and non-CF human large airway explants after 14 days of culture by measuring short circuit current in oozing chambers. As expected, we saw a decrease in chloride secretion after activation of CFTR with forskolin in the CF tissues. We also saw a decrease in the inhibition by CFTR inhibitor 172 in our CF tissues. Notably, we didn't see any difference in ENaC activity as measured by response to amiloride. Hyperactivity of ENaC in cystic fibrosis is a known feature, and it has been seen in freshly excised nasal epithelia as well as ALI cultures. Future work will be required to determine how long-term culture on gel film affects ENaC expression and function. Even so, we determined that CF and non-CF LAE exhibit genotype-specific differences in electrophysiology at day 14. Animal models of cystic fibrosis have provided key insights into CF pathogenesis and are vital to the preclinical development of therapeutics. Thus, we sought to determine if the explant system would permit long-term culture of rabbit tracheal explants from wild-type and CF rabbits. Rabbit tracheal explants, or RTE, were prepared and cultured by the same method as human explants. First, we looked at the histology of RTE at day 14 and observed a well-ciliated pseudostratified epithelium with no underlying submucosal glands. This is a known feature of the rabbit model. We also observed robust CFTR expression, staining in a diffuse pattern in RNA-scope. These observations closely align with recent work characterizing the in vivo rabbit trachea from Zhu et al. From this, we concluded that rabbit tracheal explants exhibit normal histology and robust CFTR expression after 14 days in culture. Next, we measured mucociliary clearance by tracking fluorescently labeled beads that were aerosolized onto the surface of rabbit tracheal explants. This video was taken through a dissecting microscope, and in it you can see streams of mucus flowing across the surface epithelia with bright dots indicating fluorescently labeled beads. We observed nice mucociliary clearance in RTE tissues at day 7, 14, and 21. Thus, ex vivo airway tissue could provide a nice model for studying mucociliary transport. We next assessed the electrophysiology of wild-type and CF rabbits, rabbit tracheal explants at 14 days. We observed the expected decrease in forskolin and inhibitor 172 responses in the CF rabbit tissues. However, once again, we did not see any difference in amiloride response. Rabbit tracheal explant electrophysiology was noticeably different from published measurements of fresh rabbit trachea. Most notably, amiloride and forskolin responses were far higher in the explant system compared to the freshly excised tissues. We do not yet understand what causes this difference. However, the increased forskolin response in rabbit tracheal explants could prove useful for future applications of tracheal explants in gene or cell therapy studies, where CFTR functional restoration could be used as a measurement outcome. Given the ongoing COVID-19 pandemic caused by the SARS-CoV-2 virus, we were interested to see if human explants could be used to study viral pathogenesis. Recent collaborative work from Ralph Barrick, Rick Boucher, and our lab published a gradient of SARS-CoV-2 infectivity along the proximal to distal respiratory axis. This work also established that SARS-CoV-2 selectively infects ciliated cells in the bronchi and bronchiolar regions. Thus, we infected human large and small airway explants with SARS-CoV-2 after 25 days of culture on gel foam. By immunofluorescent staining, we observed infection of alpha-tubulin positive ciliated cells in human large airway explants. The infection pattern was comparable to in vivo human tracheal sections from COVID-19 autopsy. Human small airway explants also demonstrated a ciliated cell tropism with less infectivity than large airway explants. We considered these results as good proof of concept that human airway explants could model SARS-CoV-2 infection. We then looked at total RNA copies of SARS-CoV-2 nucleocapsid genes by performing quantitative real-time PCR and comparing to a standard curve. We observed high copy numbers of the nucleocapsid genes N1 and N2 in both large and small airway explant tissues. We then assessed the relative gene expression of two interferon-stimulated genes, ISG15 and MX1, by quantitative real-time PCR. The normalized fold change in gene expression was higher in virus-infected tissues compared to mock, and noticeably high in the infected small airway tissues. However, I do want to highlight that these experiments are preliminary and have only been performed in one donor so far. Even so, this work provides proof of concept that large and small airway explants could be used to study the host gene response to SARS-CoV-2. In conclusion, we've developed a method of long-term large and small airway culture for human and rabbit. Human large and small airway explants exhibit in vivo-like morphology and maintain electrophysiologic properties for at least 14 days. CF and non-CF LAE exhibit the expected genotype-specific differences in electrophysiology. Rabbit tracheal explants exhibit normal histology, robust CFTR expression, and mucociliary transport. Further, wild-type and CF rabbit explants exhibit genotype-specific differences in electrophysiology at day 14. Finally, human large and small airway explants provide a model to study SARS-CoV-2 tropism and host gene response. Some future directions of this work I would like to better understand the advantages and limitations of this culture system by comparing human large and small airway explants to in vivo tissues by transcriptomics. It would also be interesting to study mucociliary clearance dynamics in human LAE and SAE. We need to repeat our SARS-CoV-2 infection studies with more donors. And finally, our model would be very uniquely useful for assessing the long-term fibrotic effects of SARS-CoV-2. We think this new model of ex vivo airway tissue has many possible applications relevant to cystic fibrosis. First, airway explants could be used to deepen our understanding of airway physiology, pathology, and response to stimuli. Explants could also be extremely useful as we look forward to the development and translation of gene and cell therapies. First, explants could be used to assess the tropism and efficiency of gene therapy vectors. They could be used to model the delivery of barriers to gene and cell therapy, including the mucus layer and mucociliary clearance. And finally, airway explants could provide a step between in vitro studies and more costly in vivo studies. Further, the explant model, using the explant model, you can increase the number of conditions that are studied per animal by dissecting multiple explants per trachea. For example, from a single rabbit trachea, we can usually get about 12 explants. With that, I'd like to thank the following people, especially Kenichi Okuda for his help in developing the project and dissecting explants. I'd also like to thank Barb Grubb and Troy Rogers for their help in the electrophysiology studies, and Scott Randell for his ongoing mentorship. I'd also like to thank the Marsco Lung Institute Tissue Procurement and Cell Culture Corps for their help in providing lungs and our funding sources. With that, thank you for listening, and I look forward to the discussion in the live event.
All right, excellent. Thank you, Rihanna, for that very great talk. And it was interesting to learn about this new model, which I know is interesting to many people in the field. So we have a number of questions related to what you presented. And one of the first questions from the audience is, do you have a reason or speculate a mechanism for the relatively low CFTR inhibitor effect in your healthy human explants?
Thank you for the question. I don't think we have a strong feeling as to why that's happening. I think the first Golan response and the inhibitor 172 response correlate with each other, though inhibitor response is a little bit lower. But again, we don't know exactly the reason for that. It does seem like the bumetanide is catching whatever leftover current the inhibitor didn't stop.
Exactly. So another question is, have you tried adding CFTR modulators to the gel foam sponge to try to see if you can rescue CFTR defects using your explants?
Yes, that's also very interesting. We've tried this once with an explant from nasal turbinate. Unfortunately, I guess I should say fortunately, the supply of CF lungs has decreased since the approval of Trikafta. So we actually don't get as many CF lungs these days. We were able to try it once in nasal, and we did in fact see some increase in first Golan response with the CFTR correctors. But again, this was one donor. So if we get more CF lungs, maybe we'd be able to repeat that.
Excellent. So Brian Button is asking about the submucosal gland response. Have you actually been able to see mucus be expelled from your explants if you apply a cholinergic agonist like methacholine or acetylcholine?
We haven't tried that yet, although that is on the agenda. I have seen accumulation over time, just sort of static accumulation. After about 28 days, there's a very visible mucus plug that you can kind of pull off with a pipette, but we haven't tried stimulating that mucus secretion. That's a good idea, and I think that will be done in the next few weeks.
Great. So another question from Daniel Cook is, what native immune cells do you have present in your explant culture?
So we've done some staining for CD45, which of course is just a pan-myeloid marker, and we do see CD45 staining. I haven't gone any deeper than that yet. There's sort of a lot of immune cell populations we could chase down. So I'm interested to see that as well. But as of right now, I don't have an answer.
Sure, great. So sort of a related question is, do you know whether if you were to supply exogenous immune cells, would they be able to interact with your explants?
That is a very good idea. We haven't done that yet, but that I imagine they would be able to. They seem to respond to some other stimuli like cytokines. So they seem to be responsive. It would be interesting to try that out.
Great. So this is more of a technical question related to your model, and it's two parts. So the first question is, have you tested other media for your airway cultures? And can you get them to survive past 14 days?
So we have not tried other media. I was surprised by how simple the media is and how well it seems to maintain. But we can get beyond day 21. 21 is just usually where we stop for an experiment. But I think the farthest out we've gone with the rabbit explants has been more like 41 days. And again, that wasn't a limit. That was just where we chose to do an experiment. And I think with human, it was probably about 35 days. So again, we haven't quite reached that limit yet, but it probably does exist.
So a few more like technical related questions. So typically with explants, they're a little bit thicker tissue, and then sometimes you have to be concerned about oxygenation levels or hypoxia. So have you considered potential for changes in oxygenation throughout the explant, and have you done a tunnel stain to identify cell death?
I have not done that yet. That is a very good point. We do, I guess, just on the note of tissue thickness, I'll dissect the tissues differently for different experiments. So it's possible to get the explants to be pretty thin, especially for electrophysiology. But if you're interested in submucosal glands, you might want to leave a little more submucosa underneath. So I haven't noticed any distinct cell death, but a tunnel marker would be good to study.
Great. So another, what would you say is one of the biggest challenges with this approach? So is contamination a concern or anything like that?
Surprisingly, no. We haven't had much contamination. And since these are primary tissues, we do treat with some antibiotics and antifungals immediately after dissection for the first 24 hours. I think probably the biggest challenge I've faced is the tissues are opaque. And so you can't do simple light microscopy. It's as of right now, we haven't worked out a way to do any kind of live imaging of these explants. So if you're interested in something, you kind of have to fix the tissues and do some staining.
Excellent. So another question actually from our co-chair is, have you tried any wound healing experiments with your explants?
Yes, we've done some very preliminary work on that, in particular using polydocanol, which is a chemical agent to promote injury. As of right now, we are working on characterizing the time course of that injury and even dosing. We have that worked out in ALI cultures, but I'm predicting that'll be pretty different for this tissue model. So some preliminary work and hopefully some future results on that.
That was my mistake. That was from Esther Vladar in the chat. But
my apologies.
No, my bad. Could I ask a quick follow-up, Brianna? I was wondering, this could be like a really interesting model of just studying human airway epithelial regeneration that might be more physiologically relevant than dissociating cells into suspension and expanding them and differentiating them in ALI. So have you looked carefully at the early stages during this, possible injury period where you don't see CFTR current to see what's actually happening to the epithelium?
We have not. We've really only done electrophysiology, and that was our marker of tissues not being up to full health. But it would be interesting to do some fixing and staining and see sort of what's happening there, if there are tight junction loss or anything like that.
All right, excellent. So thank you so much, Rihanna. That was a very great talk and excellent discussion. Thanks to the audience members. And now we will move forward to our next talk.
My name is Margaret Mikicic. I'm a graduate student in Craig Hodges' lab at Case Western Reserve, and today I will be talking about 3 new W1282X CF mouse models for examining in vivo nonsense mutation correction. There are no relationships to disclose related to this presentation. Not all mutations can be treated by the same means. Therefore, it is beneficial to have a variety of nonsense mutation models. Now, one can study in vivo correction in a W1282X specific model. We have created 3 different models using CRISPR-Cas, one with a W1282X in mouse CFTR, one with W1282X in human CFTR, and a reporter that upon W1282X correction should produce fluorescence and bioluminescence. The ideal model will depend on your question, but all are powerful tools that can be used to study pharmacological, gene editing, and other novel W1282X treatments. The mouse W1282X model contains the W1282X mutation in mouse CFTR. This is a typical CF mouse. All of the mice I'll introduce today are on a black 6 background. Homozygous mouse W1282X mice show reduced mRNA expression compared to wild type, which is normalized to 100%. When compared to an already published model for G542X in mouse CFTR, mouse W1282X has similar mRNA levels as expected. Here, we measured change in electric potential across intestinal and nasal epithelium of wild type and W1282X mouse tissues to test CFTR function. On the left, we have intestinal short circuit current. Mouse W1282X mice show significantly reduced electric potential compared to wild type, indicative of a lack of functional CFTR. On the right, we have nasal potential difference. Wild-type mice show a moderate negative NPD, while mice homozygous for W1282X show a slightly positive NPD, indicative of a lack of functional CFTR. Using mouse small intestinal-derived organoids, one can conduct a forskolin-induced swelling assay. Forskolin activates CFTR, allowing organoids to swell if CFTR is functional. We quantify FIS by change in area over time. Wild-type organoids with functional CFTR, this red curve, show rapid and robust swelling. Mouse W1282X organoids, this orange curve, do not show swelling, indicative of a lack of CFTR protein. G418 is an aminoglycoside antibiotic capable of reading through premature stop codons. Here we see that treatment of the W1282X organoids with G418, this green curve, does lead to moderate swelling, indicative of some rescue of the W1282X protein. SMIG1I is an NMD inhibitor, and NMD is the process by which transcripts containing premature termination codons are degraded. The combination of G418 and SMIG1I, this purple curve, yields impressive correction, though it does take several hours to reach near wild type levels. I've demonstrated these mice can be used to study in vitro pharmacological correction, but they can also be used to study gene editing. Here I'll describe how we have gene-edited CFTR in small intestinal organoids. First, organoids are dissociated to make single cells. Then they're incubated with guide RNA, W1282X corrector oligos, and Cas9. Next, cells are electroporated, and surviving stem cells are allowed to form up organoids. Next, we do a primary selection by treating the organoids with forskolin. Organoids that swell, indicative of CFTR correction, are subcloned and kept in their own well. Subclones are then allowed to multiply and form homogeneous populations from which DNA is isolated and sequenced. Here we have images of a homogeneous population of mouse W1282X organoids, all derived from a single edited swollen organoid. Here they are treated with forskolin, and we see widespread swelling within 1 hour, indicative of correction. Recall that unedited mouse W1282X organoids do not swell. Sequencing confirms 2 of the edited clones are heterozygous for the wild-type allele. Next, I'll introduce the creation of the humanized W1282X model. This mouse has mouse CFTR gene knocked out and a copy of human CFTR randomly inserted into the genome. It's important to remember mouse and human CFTR sequences are different. On the gene level, they have a nucleotide sequence that is 82% identical in the coding region. On the protein level, human and mouse CFTR are 78% identical and 88% similar. The humanized W1282X mouse was created from an existing mouse with wild-type human CFTR, published in Scientific Reports in 2019 by Guanas et al. This humanized CFTR mouse was made using a bacterial artificial chromosome containing the whole human CFTR gene, along with upstream and downstream regulatory regions. This construct was randomly inserted into mouse chromosome 8. Then this mouse was crossed with a mouse CFTR knockout, so the resulting mouse is null for mouse CFTR and positive for human CFTR. This humanized mouse expressed human wild-type CFTR in the main tissues of interest in CF and displays human CFTR function in the airway and intestine, which rescues the CF manifestations of the mouse CFTR knockout. The wild-type humanized CFTR mouse I just introduced was then edited with CRISPR-Cas9 to alter the human CFTR sequence to contain the W1282X mutation. I'll call this mouse humanized W1282X. Here I'm showing functional data from the humanized wild-type CFTR and humanized W1282X mouse. Wild-type, in this case, refers to mice with wild-type CFTR in the mouse genetic sequence, no transgene. On the left, we have intestinal short circuit current. Humanized CFTR with wild-type sequence does not display the same level of correction as wild-type mice containing the mouse wild-type sequence. However, humanized W1282X mice display significantly reduced activity indicative of lacking functional CFTR, acting here like a typical CF mouse. On the right, we have nasal potential difference. Humanized wild-type mice do show CFTR function, albeit slightly reduced compared to mouse CFTR wild-type. Humanized W1282X mice display a slightly positive NPD, indicative of lacking functional CFTR, again, typical of a CF mouse. Together, this suggests that the humanized W1282X mouse made with the transgene still behaves like a typical CF mouse in terms of CFTR function. Pharmacologic and gene editing correction of the humanized mouse is currently being studied in organoids. Finally, I'll introduce a third W1282X specific mouse. This mouse is a reporter that when treated with an agent that corrects the stop mutation should yield fluorescence and bioluminescence. This construct contains human exon 23 with the W1282X mutation, followed by P2A, which is a self-cleaving peptide sequence, followed then by sequences for M. venus and acyluciferase, and then a polyadenylation sequence. The reporter mouse construct is at the ROSA26 locus under the control of the CAG promoter. Under normal circumstances, the W1282X stop mutation prevents any products from being produced. When W1282X is corrected, an exon 23 peptide is produced, as well as fluorescence and bioluminescence. Here we see what could be visualized if gene correction were successful in every cell of the reporter mouse. This is not the reporter mouse. The transgene positive mice here constitutively express acyluciferase and M. venus. And these are live sedated mice imaged by an IBIS system. And only after 10 minutes of acrylamide treatment do we see bioluminescence throughout the whole mouse. Here, using the same system, we visualize the constitutively expressing M. venus and oculluciferase mice under fluorescent conditions. So these are the types of results that one could hope to see following successful whole body gene editing in the reporter. But again, this is not the reporter. We don't quite have this data yet in vivo for the reporter, but we do have some preliminary in vitro data. Here we visualize small intestinal organoids derived from this reporter mouse. And these organoids are controls that were not subjected to treatment. And here we only see background fluorescence. Here, organoids from the reporter mouse were treated with guide RNA, Cas9, and a W1282X corrector oligo. And we do see stronger signal corresponding to a couple organoids. Sequencing results showed that the organoid cultures treated with Cas9 and guide RNA alone had 0% wild type sequence as expected, whereas cultures treated with guide RNA, Cas9, and the corrector oligo showed 6% of sequences contained wild type. Therefore, some correction is occurring. Now we need to confirm that the corrected organoids are in fact those displaying fluorescence. In summary, 3 novel W1282X specific mouse models have been created. The mouse and humanized W1282X strains are available for research, and the reporter is still being characterized. Both mouse and humanized W1282X models show decreased CFTR mRNA, growth, and electrophysiology, typical of other CF mouse models. Moderate in vivo mouse W1282X correction via pharmacological treatment has been demonstrated, as well as in vitro mouse W1282X and reporter correction via gene editing. Pharmacologic and gene editing correction of the humanized W1282X mouse is ongoing, as are studies with the reporter. These are strong tools available for research to study in vitro and in vivo correction of the W1282X specific mutation. I'd like to thank Emily's entourage for funding this work, the CF mouse models and transgenic and targeting cores at CASE for their help in creating and maintaining these strains, and the CF core is supported by the CF Foundation, and I am supported by the Functional Genomics Training Program. Thank you.
Hey, Margaret, for that great talk, and apologies to the audience for the unexpected delay in the slides compared to the audio. So that's unfortunate, but still interesting work that we just got to hear about. So Margaret, we do have a few questions from the audience, and one of those questions is related to if you have a predicted or a proposed way to best administer new genes and editing vectors to inflamed airways. Do you have any ideas about that?
I do not. We haven't done a lot of in vivo gene editing work quite yet. We've really stuck to the intestinal organoid models. But you know, in vivo and looking at the airway, it's obviously very important for CF, so it is an interest.
Great. So related to, in your beginning of your talk, you showed that there was a time delay for correction when you use G418 and SMG1I, I believe. And do you have a speculated mechanism for that?
We do see that the W1282X organoids show slower correction, and we have some further data that shows that correction continues even at like the 24-hour mark. And this is different than some of the other genotypes that we've looked at in organoids. And so we're not completely sure why that is yet. This is something that I'll continue to look at and also see if the speed of correction can be altered with the addition of CFTR correctors, which might be the case. So it is an interest, and I'll be investigating these.
Great, great. Also, in the beginning of your talk, when you showed the gene editing experiments for your intestinal organoids, you might have stated it and I missed it, but how much correction did you have for those organoids?
So the mouse,
Yes.
W1282X, so we confirmed, we only took a select few number of clones that displayed swelling. And so we had 2 clones that we did get wild type sequence in by sequencing. And then when we created cultures from those wild type clones, it's near 100% correction within that homogeneous population.
The expanded population.
Yes.
Okay, great, great. And then another sort of related question, a lot about the timing and stuff. So when you showed correction for your in vivo assay, where I think you're using bioluminescence or some other in vivo marker, I unfortunately was hard to follow along, but that correction seemed much quicker compared to your organoid data. And so again, do you have a speculation about the difference in those timing events?
Yeah, so to clarify, the images using the IVIS system of the live mice with oculoluciferase and M. venus expression, those are not the reporter. They are different reporter mice that constitutively and ubiquitively express M. venus and oculoluciferase. And so that's an example of what you could see using the W1282X reporter. But you're right that in those mice, even just 10 minutes after oculolamine treatment, you do see widespread bioluminescence. And then in the fluorescent mice, again, you see very quick. But I would like to just clarify, those are not the W1282X reporter. It's an example of what you could see with this reporter. So those experiments are ongoing. The mouse is just still new.
All right, great. Thank you for that clarification. So another question then, again, maybe this is not a great question based upon the fact that I'm not getting a lot of the facts straight about your talk. I apologize for that. But at the end, you showed 6% correction in your organoids. Is that correct?
Yes. So the image that is shown is of two organoids that are fluorescing. Separately, cultures of organoids were sequenced, and that did show 6% wild type sequence. The image does not correspond to the culture that was sequenced, and so the next step is confirming that the organoids that we see fluorescence in are those that contain the wild type sequence. But you are correct that we did achieve 6% wild type sequence.
Okay, great. So I believe our co-chair Finn Hawkins has a question for you.
Margaret, that was a really nice talk, and apologies to you and the audience for the technical difficulties. I wondered, maybe you might just summarize your future plans, because the disconnect between the audio and the video, I think, robbed you of that possibility, and it'd be nice to hear you articulate those again.
Yeah, so the mouse W1282X model, I feel the story is pretty complete. We've shown in vitro correction and gene editing correction. The humanized mice, I'm interested in looking at those organoid studies in forcible and induced assays to see, we would expect similar response, but we know that some of the drugs respond differently, mouse CFTR and human CFTR. And then the reporter W1282X mouse is still pretty new, and so I'm excited to see if we see bioluminescent and fluorescent correction as we do in the example mouse that I showed pictures of.
So if there are no additional questions, again, Margaret, thank you for that talk. Unfortunate for the technical difficulties, but the work was excellent. We appreciate you taking the time to discuss it with us. So I think we will move forward to the next talk.
Good morning. I'm Xiaoming Liu from the University of Iowa. Today, I'm going to talk about genetic editing in ferret airway epithelia made by CRISPR nucleus delivered with amphiphilic peptide shuttles. I have nothing to disclose in this study. Despite the development and use of CFTR modulator in CF disease treatment, the recent emergence of the CRISPR-Cas9 made gene editing showed ability to correct CFTR function in human airway epithelial cells in vitro, suggests the potential for permanent treatment of CF disease using the gene editing approaches. However, the efficiency of the delivery of the CRISPR nucleus complex into the cell is very challenging. To this end, the Phaedon Therapeutic recently developed amphiphilic peptide shuttle for protein delivery into the cells. The shuttle was a short cell-penetrated peptide and in the thermal leakage domain that engineered to perform highly effective and safe cytoplasmic delivery of multiple types of therapeutics, including the proteins. Peripheral studied in the Dr. Paul McQuaid's lab and the University of Iowa has demonstrated amphiphilic peptide enable delivery proteins and the CRISPR-associated nucleus to the human airway epithelium in vitro and the mouse airway epithelium in vivo, and the result in the gene collection, gene editing. However, the mouse CF model lack of the lung disease phenotype and cannot be used for the gene editing study for CFTR function collection. To this end, the CF ferret model developed by Dr. Inhofe at the University of Iowa, could develop both of the lung and pancreatic disease like humans CF disease. In addition, a transgenic ferret model was also generated in this lab. This P reporter ferret in harboring a transgenic cassette with membrane-bound tomato and membrane-bound EGFP in the presence of a clearly combinase or disrupt the LOXP locus will result in the simultaneous conversion of the tomato to the GFP fluorescence. Using the ferret model, we first can be tested the amphiphilic peptide shutter for delivery proteins into the airway surface, airway epithelial cells in vitro by using the polarized airway epithelial cultures, and also in vivo using the Y-type ferret lung. And for gene editing studies, we used the Rosa tomato GFP reporter ferret. The airway epithelial generated from this model or the lung of this model can be used for delivery of the CRISPR nuclease and the guide RNA to LOXP. Following the gene editing, the conversion of the EGFP in the epithelial cells can be detected and saved as the index of the gene editing. In vitro study demonstrate the GFP nuclear fluorescence can be efficiently transfected into the polarized airway epithelial cells of the ferret, as seen in the nuclei of the epithelial cells. And the delivery of the Cas9 guide RNA complex or CPF1 and the guide RNA to LOXP complex, resulting in the conversion of the EGFP in the polarized epithelial cells of the ferret. In vivo study using the 2-week-old of the Y-type ferret, the GFP delivery made the amphiphilic peptide shorter made the EGFP delivery can be observed in the intralobar airway. The efficiency of the gene protein delivery in the airway, polarized airway, was about 9%, and in the juvenile intralobar airway is about 16%. Gene editing efficiency, in vivo gene editing efficiency for CRISPR-Cas9 is about 2.3%, and for the CPF1 made gene editing is 0.9%. In vivo study for the biodistribution and the efficiency of amphiphilic peptide shuttle made the peptide transfection. Using the adult ferret, the DRI fluorescent labeled peptide was intratracheally delivered into the ferret lung, and the trachea or the lung was evaluated for the fluorescent labeled peptide in the epithelial cells. As seen in above the trachea and in the intralobar bronchial airway epithelial cells, all have an abundance of the fluorescent labeled peptide in the epithelial cells. Quantifications demonstrate about 54% of the trachea epithelial cell can be transfected with the fluorescent labeled peptide, and 45% of the intralobar airway epithelial cells was transfected. In the juvenile ferret airway lung, the biodistribution efficiency was also investigated, with a similar result seen in the adult trachea. The fluorescent labeled peptide was transfected into the trachea and the bronchial airway epithelial cells. Quantification study demonstrated about 48% of the tracheal cells was transfected, and 27% of the intralobar airway epithelium was transfected. To identify which epithelial type was transfected with the amphiphilic peptide shuttles, immunocolocalization with epithelial marker, cellular marker alpha tubulin, and the goblet cell marker, NACFC was performed. As demonstrated in these images, both of the cellular cells and the goblet cells can be efficiently transfected with the amphiphilic peptide shuttles. The efficiency of the transfection in the adult trachea is about 7% for the cellular cells and about 4% of the secretory cells. In the intralobar airway, 47% of the cellular cells and 29% secretory can be efficiently transfected. Similar in the juvenile trachea, 16% of cellular cells and 7% of secretory cells was transfected, and 7% of cellular cells and 8% of secretory cells can be transfected in the juvenile ferret intralobar airways. Gene editing studies in the LOSA reported transgenic ferret demonstrate the efficiency GFP, gene-edited GFP cells was observed in the trachea of the LOSA animal following the delivery, following the amphiphilic peptide delivery of CRISPR-Cas9 complex. This GFP cell was clearly in the epithelial layer of the trachea. Similarly, the GFP cells in the bronchioles and the intralobar airways. The efficiency of the gene-edited cells in the trachea is 4.7% and about 4% in the intralobar airway of the adult ferret. Similar study using the identical approach, we found the efficiency of gene editing in the juvenile ferret is less efficient. However, the GFP cell also can be found in the epithelia of the trachea and the intralobar airway. The gene editing efficiency is about 3.3 in the juvenile trachea and about 2.8 in the intralobar airway epithelium of the juvenile ferret. Colocalization, the gene-edited cell epithelial cell type using immunocolocalization methods. We also found both of the cellular cells and goblet cell can be genetically edited in the ferret airway epithelial cells using the amphiphilic peptide shuttles. The efficiency in the adult trachea, about 1.9% of cellular cells and 2% of secretory cells in the adult trachea can be genetically edited. In the intralobar airway of the adult ferret, there was about 2.4% of cellular cells and 1.3% of secretory cells can be genetically edited. Interestingly, we also observed a much more abundant genetically edited GFP cells was observed in the distal airway. The distal respiratory airways, this GFP particles is up to 4% in the alveolar region. In addition to the able to mediate CRISPR-Cas9 to gene editing, we also test whether there's endophilic peptide shuttle could mediate CRISPR MET7 gene editing in fed airways. As expected, both of the adult ferret and the juvenile ferret airway epithelium can be gene edited by CRISPR MET7 nucleases delivered with the endophilic peptide. Also, the efficiency is less compared to the CRISPR-Cas9. In summary here, the fed and endophilic peptide shuttle can efficiently transfect the proteins into the ferret airway epithelial cells. The CRISPR nucleus GFP guide on RNP transfect with the endophilic peptide shuttles demonstrate the ability of gene editing in various epithelial cell types with a comparable efficiency in airways of adult and juvenile ferret, improving the gene editing efficiency, particularly airway epithelial progenitor stem cell subpopulations is important for the therapeutic potency of gene editing for CF disease in future. Finally, I would like to take the opportunity to thank Dr. Engelhardt and the member in his lab for their assistance and support in this project. Thanks, David Guy's group at Ferret Therapeutic for their support and assistance in this project. And thank you for your attention. With that, I would like to take questions.
All right, great. Thanks so much, Xiaoming, for that great talk. And now it's time to start a discussion with some questions from the audience. So one question from our audience member is, are basal cells targeted by your peptides?
Oh, thank you. By my just observation, I cannot say it's not, but we didn't see any in vivo. We didn't see any like targeted, even like transfected or by the peptide, endophilic peptide. So maybe we need to do more like experiment or like do more like analysis, but at least the efficiency is very, very low.
Okay, great. So Dan Weiss is asking, how do you propose to best administer the new gene transfer and editing vectors effectively to inflamed or injured airways?
That's a good question. And we think about it use like injured airways. But the first we need to like overcome, try to assess why the efficiency can improve, how the efficiency can improve by the penetrate the mucus layer of the adult trachea, particularly in the adult trachea. So we tried to like use like EGTA or try to call like transfect with not use like a peptide, but use the other like dye or delivery. And the efficiency actually is not succeed like a significant improve in the ferret, although there's some data or reported in the mouse area, but not, we didn't see any improve, at least by our like 3 times attempted try to improve is not. So maybe this is a challenge for us also, how to improve the gene delivery in the ferret lung.
Great. So a related and follow-up question is from Scott Randall, and he's asking, have you considered mild injury before adding the Cas peptide in order to target the basal cells?
Yeah, that's, thanks, Scott. Yeah, this is a really like a great idea. We are thinking about it. And first, actually, we are trying to see in the juvenile airway might be getting more like easily to assess the basal cells rather than the adult. And however, the delivery for the in the juvenile airway ferret, because it's small, cannot use any like have to use the like surgery or surgical approach. So we currently try to, because by the right now we use the intratracheal installation for delivery. The efficiency is lower because when we see the in the histology, but I don't think it's because of the transfection, it's because of the delivery. Maybe, my guess, we need like next step, we might be trying to use the surgical delivery the lung, might the juvenile ferret, like 2-week ferret lung, might be getting improve the gene editing in the basal cell.
So related to that, you did show in some of your data that the juvenile ferrets had higher efficiency compared to the adults. Do you have a suspected mechanism for that? Is it simply a delivery element, or is there some other biological factor influencing that?
I think based on our observation, the editing efficiency in the tracheal, the proximal airways, is like more efficient than in the distal air. We think about this, I think it's maybe it's a cell biology difference in the like airway tree, maybe cause the gene editing efficiency, or is the from the efficiency of delivery by the peptide delivery use the DR. I think this is not like because delivery possibly caused by the efficiency of the gene editing in the different cell type or the biology of the airway epithelia from the proximal to the distal maybe have some like heterogenesis.
Excellent. And another related question is, you did show in some of your data a slight trend for increased efficiency in ciliated cells compared to secretory cells. And again, do you have any speculated mechanism for that?
If you look at the population, because the population and in the main epithelial cell type in the airway, say there's more abundant than the other cell type. I think we don't know the tryptosome of the gene editing at that point, because we might be, if you look at the percentage of the compared to the total cell number epithelial cellular cell to the edited cell type, maybe that is not see any like difference, significant difference. I think that the cause probably because of the population in the airway might be caused currently the data difference.
Excellent, thank you.
That was beautiful talk, and those tools are very, very exciting. I had a couple of questions. One gets back to a question Dan Weiss asked earlier, which is, gene editing in vivo is hard enough. How are we going to gene edit the CF inflammatory airways? So I guess I'm wondering, have you had a chance to look yet in the CF ferret to start getting a sense of how much more difficult it's going to be to target the airways in a CF animal or human?
Yes, I think the first thing we are thinking about is we have a CF model, ferret model, and we also breeded the CF model to the Lhasa animal background. By our observation, there's enrichment because some cell type maybe have like a hot spot for gene editing. If you see the LOXP site was edited or indexed, most likely like 70 or 80% of the cell was also edited by the other cell type. There's a kind of gene editing like enrichment. So we can like assess the editing event by use the reporter and as well as the CF. I think we can try the like nasal epithelial or proximal epithelial, because the function, we need to actually how the efficiency we need to like test in the airway culture first before moving to the in vivo study.
Okay.
There's a long way to go, I think.
Yes, great. Thank you very much for the talk.
Thank you.
All right, great. I think then we will move forward to our last talk of the session. Thanks again.
Thank you, the North America Cystic Fibrosis Committee, for inviting me for this workshop presentation. And thank you everyone who's watching. So the title of this work is the F508del and G532X sheep models exhibit a severe cystic fibrosis phenotype, and the tracheal epithelial cells isolated from those animals, they respond to human therapeutics in vitro. Before I start off, I would like to say there are no relationships disclosure related to this presentation. So I'm going to start off this presentation saying how we produced the models containing the F508del and G532X mutations. So basically, these two mutations are the most common mutations in the North America cystic fibrosis patients. And we use the CRISPR-Cas9 system along with the ssODN, which is the single-stranded oligonucleotide, designed to contain the mutation that we desire. So we use the system to target the exon 11 in order to introduce the F508del mutation. And for the G532X mutation, we design another CRISPR-Cas9 with another ssODN to target the exon 12 in order to introduce that mutation. So here is a representation of how the mechanism it would work. So basically the ssODN aligns against the double-strand DNA break where CRISPR attacks, and the cell repairs the DNA based on that sequence. So we have the confirmation here in the second panel of the cells that contain the biallelic F508del and cells that contain the G532X mutation. So if you see here, the CTT was deleted in those cells with the F508del mutation, and the cells containing the G532X, the codon GGA was changed to TGA. We changed from glycine to a stop codon. So then we used these cells that we produced in the somatic cell nuclear transfer, in which we transferred a single cell into a enucleated oocyte that we previously enucleated the oocyte from a oocyte donor. And then we do diffusion and activation, and this reconstructed embryo, it is transferred into an ester-synchronized surrogate mother. And in sheep, the gestation is about 5 months. So after 5 months, we have those offspring. So the objectives here was to identify the histopathological features associated with the CF disease in both lambs with those mutations, and also evaluate the human therapeutics to restore the CFTR channel in the sheep tracheal epithelial cells. So here is the intestinal tracts of the wild type F508del sheep and the G532X sheep. And you can see here the difference between them, that you have this portion of the small intestine dilated because we have an obstruction here in the intestine. And that's why it's called this small intestine here. And also this spiral column, it's quite small compared to the wild type. So in these histology slides, you can see the wild type is empty, and we have a quiet space between the cells. And here in the F508del and G532X lambs, they have this thick mucus inside of the intestine, which causes the obstruction. And for the column, it happens the same way. For the wild type, we don't have the mucus accumulation, but for the F508del and G532X, we do have the mucus here. And the meconic ileus, which is this abnormality here, it also happens in the CF patients at the birth, but the frequency is about 15% of the infants. But in sheep, all of these F508del, G532X, they are similar to the CFTR knockout sheep that we also produced. And these are only related to a very severe phenotype seen in some humans. So for the pancreas, it's another abnormality that also happened in humans. we have the damage of the pancreas. So in these slides here, we have the atrophy of the areas of the pancreas, of the exocrine areas. And also you can see the fibrosis forming here in between the cells. And also you can see here the acini dilation. So these ducts, they are quite dilated. And this causes the problems with the pancreas. So you don't have more of the pancreatic juice being produced in this pancreas, which cause some problems associated with the CF disease. And for the liver, we have here a comparison of the three livers, the wild type, the F508del, and the G542X. So you can see in the wild type, it looks quite healthy, and also the gallbladder is present there. But the first thing you can see is that the gallbladder is here in the F508, it looks absent. And here in the G542X, it's quite small. And one thing we can see here is the peripheral fibrosis, which is this thick amount of cells here around of the ducts. And also the cholestasis found inside of the liver. So at the moment of the necropsy, we isolate a piece of the trachea for isolation and differentiation of the epithelial cells in permeable supports. And then we use them in the using chambers for electrophysiology assessment. And here is the representative trace of the short circuit current that you can see for the F508del mutation cells. We introduce the amiloride that cuts off the short circuit current. And then we add the forskolin. And from the forskolin, it's going to activate the CFTR channel. And then we add the VX770, which is the potentiator from the vertex. And then the genistein is also a CFTR activator. And the GLH101 cuts off the power from the CFTR channel. And then we stop the reaction. So using these three different treatments, which are the current hemotherapeutics that we have, the tezacaftor and the elexacaftor, they had a better response in those cells. So basically what's going on here is that F508 mutation, it prevents the protein to be properly folded, and then the protein ultimately is degraded by protease instead of the epithelial cells. So those compounds, they work as a chemical chaperones, and they help the folding process of the CFTR protein. And after that, the protein is able to go to the Golgi complex and ultimately goes to the membrane to work and pump the chloride ions. So here we tested a similar reaction with the VX tezacaftor and the elexacaftor along with the potentiator, but also we did the combination of the VX61 with the elexacaftor together along with the potentiator. And then this is also a combination that is commonly used in that medication, the Trikafta, and the response containing those combinations, it was much better than each one of them along with the potentiator. So for the G542X cells, we used a nonsense mediated decay inhibitor, which is the SMG1, and also the G418, which is the gentamicin antibiotics. And also we tested it with the tezacaftor here. And the response was not as great as it was expected, as the SMG1 inhibit the surveillance pathway. So we had some response here compared to the MSO reaction. So what's going on here? Basically, with the nonsense mutation, we have the mRNA decay inside of the cells, and basically we don't have the production of the protein, and the mRNA is shorted. So this what we do here is blocking the surveillance pathway of working in order to prevent the mRNA decay. And also with the gentamicin, that's a read-proof drug that we use. They should be prevent the stop codon of working. So the ribosomes just go through the reading, and then we should have a little bit of the protein being produced and going to the membrane. So in summary, we can say that we successfully produced 2 additional CF sheep models carrying the 2 most common mutations of the North American CF patients. And also they exhibit very severe phenotypes that were seen in the CFTR knockout animals, and also in severe cases of the human patients. And the severe abnormality was associated with the CF disease. And like I mentioned, the meconic ileus is right now one thing that prevents them to live after birth for a long period of time. And the alpha-V-Del trachea epithelial cells, they respond to the current human therapeutics to restore the CFTR channel activity. And for the G542X epithelial cells, they had a modest response to human therapeutic to restore the CFTR channel. And I would like to say my acknowledgements to all the authors who contributed to generate and characterize the alpha-V-Del and the G542X chip models. Also, the epithelial core cell core at the Case Western Reserve University, CF Center, and Dr. Bridges at the Rosalind Franklin University for the donation of the SMG1 inhibitor. And also would like to mention that this work was partially supported by the U.S. Department of Agriculture multi-state project, and also the Utah Agricultural Experimental Station. So with that, I would like to say thank you all for watching this presentation, and I'll be open to questions now. Thank you.
All right, thanks Yuri for that really great talk. A lot of work just presented there. I know how much effort goes into making large animal models. So we're going to transition to some questions from the audience, and I will just start it off. Do you have any plans? You mentioned the meconium ileus is a concern for the model, which is not unexpected. And so are there any plans for your group to make a gut-corrected sheep model?
Yeah, sure. That actually one of the things we are currently working on is producing a plasmid vector transgene containing a intestine-specific promoter, so we can address that problem. And one of the ones that we are trying is the IFABP, which is intestine fatty acid binding protein promoter, which has been used before in pigs. And also we are trying with the bilin, which is a big promoter, but it's very specific for intestine, and the LFABP, which is similar to the IFABP, but that one should be specific for liver and intestine as well.
Excellent. So we have a question from Scott Randall, and he's asking, can you give any idea about the % of wild type CFTR correction that was observed in the Delta F508 and the G542X models?
So, sorry, can you say that again?
Sure. So the question was, do you have an idea of the % of wild type CFTR correction for the models that you showed us?
So basically, when we introduced the F508 and the G542X, we had, for the knockout, it was the biggest portion that we had initially. It was over 90%. And for the introduction of each of the mutations, we got about like 9% for the F508del and 15% for the G542X. And we have also tried in vitro to do the opposite, to see how it would work to fix the mutations. And that would be something similar, because with the knockout, with CRISPR-Cas9 attacking and breaking producing the double strand break, it's almost inevitable to not have the knockouts effect. So usually the average is about like 10%, 15% that you can get to introduce a mutation or just correct that mutation.
Okay, great. Another follow-up question from the audience is related to sheep biology. So are sheep lungs colonized naturally by bacteria?
So we don't have that data yet because the sheep, they don't survive enough to have data postnatal. So basically, at the moment of the birth, they have a very healthy lungs. But in order to observe more in the postnatal stage, we need to fix the intestinal in order to see if they're going to survive a little bit longer and see how the effects will be observed in the lungs.
Okay, excellent. So related to your CF sheep models, did you look at birth weight and size, and are there differences in those parameters?
Yeah, we have seen a little bit because of the LOS, the large offspring syndrome, but we believe that it's probably related to either one of the treatments, because we have had animals that were born with like about 10 pounds, but we also have some animals that were born with like 23, 26 pounds. So there is a quite big difference due to the LOS syndrome.
Great. And then another question is, what do you think are some of the advantages of the sheep model compared to the other models that are available currently?
So sheep as pig is very similar to human in many ways. And for example, sheep has been used as a lung developmental model compared to human for a long time. So that's an advantage. But also with sheep, we can try some different approach that we probably cannot try with like pigs, which is some prenatal therapy, because sheep produce only one offspring or maximum two, which is similar to human. So we could try some prenatal gene therapy or something like that that would help us to understand better and how we could potentially in the future fix this kind of mutation before the birth. And now also you got to consider that damage to the pancreas that occurs during the pancreas development. So it would be important to fix that problem before we have that. So sheep would be ideal to test this kind of approach and see if we could prevent damage to the tissues.
Excellent, excellent. So I think Vin might have some questions for the group.
Yeah, I was hoping to pose this to all the panelists and get your thoughts and your brief input. I mean, I think we heard the spectrum here from iPSC technology to very advanced mouse models to ferret models and sheep models. And my question to you is, do we have the tools that we need? Are there key things that we haven't made yet? And what's your perspective on our current situation? So
with the gene editing stuff, because we gotta think about like different aspects. One is the gene editing efficiency to fix the mutations that you want to fix. The other thing is how you're gonna deliver that and what kind of method you're gonna use. If you're gonna use ribonucleoprotein, or if you're gonna use mRNA or DNA as the material for delivery in order to fix something, and also the method of delivery, because it is not efficient for everything. So when you're dealing with some live organism, it's a lot of things that you need to consider. Also the toxicity of the material you're going to use for delivery. So there is a bunch of things that need to be addressed. But first, you got to at least optimize the gene editing efficiency in order to have something that's going to be feasible for later delivery. So it's a bunch of things that you got to think and organize before you move on. And of course, the safety of the animals that we are trying or we're going to try in the future, that's going to come also for thinking.
Yeah, absolutely. I see you're showing up in random orders on my screen. So maybe I'd ask Rihanna, do you have comments? And then I'll call on Junji.
Sure. I think the first thing that comes to mind is not so much a tool that's missing, but more of a, I guess, a unifying thought as we look towards like gene and cell therapy. What are we actually measuring? And how do we compare results across models? And then even just like what's the gold standard that we're comparing to? So it's not necessarily a tool, but maybe kind of discussion that other groups should kind of have all together.
Yeah, I think that's a great point. Junji, and then maybe Margaret.
Yeah, I think I agree with what Yana just said. It's really, for example, when we talk about cell therapy, the engraftment is a big issue. Like which models or what kind of cells we should use to test the engraftment ability, how the cells integrate. So I think that's a good thing. I don't know which models are good for this, but I believe there are a lot of hurdles that we need to tackle.
Great. Maybe Margaret and then Xiaoming.
Yeah, I just think that to achieve a cure for all needles, we need to ensure that the models we're creating can provide data for rare mutations as well, whether that means using existing tools or creating new models. I think that's also important to keep in mind.
And Xiaoming. Oh, Xiaoming, I think you're muted.
Mute, yeah, sorry. I'm mute. So I think currently the field will find the contribution of the submucosal gland in the CF disease. So also the discovery of the ion side and the expression of the CFTR. And we currently know the CFTR is highly expressed in the ion side and the submucosal gland, and the secretion and the maintenance of the physiology, like the mucus homeostasis. The submucosal gland contributes to a lot in the CF disease. So based on this, like an anatomic structure, I think the ferret is the advantage because the entire airway, cartilage airway, with the even distribution of the submucosal gland, just as seen in the humans. In addition, the size of the ferret is it's not huge, but it's still manageable, or it's good size for lung disease, for the gene therapy, for cell therapy, for preclinical study for development, treatment approach or strategies. So I think for the gene editing, I think the currently is encouraged, but that the efficiency is low, and also how to assist the progenitor or stem airway epithelial stem cell population is very challenging, and how to it's hard to injure the lung and do the gene editing in the clinic. I don't think it's not correctable. Maybe I think the only thing we maybe need some develop other reagent can be as the gene delivery to the basal cell is very important in future. Use any model can with the CF disease phenotype, lung disease phenotype.
Great. Okay, well, I would like to thank all of our excellent talks and speakers today. We really appreciate you taking the time to share with us your work. And also thanks to our co-chair, Finn Hawkins, who was helping to moderate all the questions so that we were having a great discussion with. And thanks to the audience for your attention. And we hope you've enjoyed this session.
Thank you.
Thank you, everyone.